Remember, there's 3 points of control within the cycle. The first step, which is the condensation of oxaloacetate with our acetyl-CoA. And remember, it's inhibited by ATP and NADH. And a downstream molecule, succinyl-CoA. I guess I didn't mention it before, but it's also product inhibited by its citrate. So if a lot of citrate's built up, it's not going to go forward. Our first decarboxylation step, we're going from isocitrate to alpha-ketoglutarate. It's activated by our molecules that are indicators of low energy reserves. ADP and NAD plus are, as we found out from last quiz, right, are molecules that are indicators of low energy reserves. And it's inhibited by high energy reserves, ATP, high levels of ATP and NADH. Our alpha-ketoglutarate dehydrogenase complex, which is our second decarboxylation, going from alpha-ketoglutarate to succinyl-CoA, it's inhibited by, again, ATP and NADH, and its product, and activated by ADP and NAD+. This isn't too bad, is it? Right? If in doubt, what's your inhibitor? ATP or NADH, right? And what's your activator? High levels of ADP or NAD+, right? Okay, I think you guys got that. Now, I don't want you to forget that outside the cycle, it's not within the cycle, so make sure that you're reading my question, all right? Make sure you're reading my question. Am I saying 3 points of control within the cycle or within and outside the cycle? It's that pyruvate dehydrogenase complex going from our pyruvate to acetyl-CoA. It's inhibited by ATP and NADH, and also product inhibited by acetyl-CoA. Now this pre-citric acid cycle I don't have necessarily on our little review sheet, so maybe you can add it on the back, right? Add it on the back because it is very important and it is under control. This is a great way to see here we need points of control, pre-citric acid cycle, going from oxaloacetate and acetyl-CoA to citrate, and our oxidative decarboxylation steps. This is where we have inhibition and their control. The starred steps are points where we need an electron acceptor. So if we don't have electron acceptors, these are not going to go forward. So if you're resting, and hopefully you guys are in a resting state right now, that you're going to have a high ATP and low ADP levels. That implies that you have, you need and you're using little energy. So the ratio of ATP to ADP is high. Also, you have high NADH to NAD+, especially since maybe you ate breakfast and you're here resting. Nice, no one's worrying about it. Cells in an active metabolic state, if you were able to get out for a run this morning, maybe not, then your ATP would be low and high ADP. That would mean low energy reserves. And then high NAD plus and low NADH also implies low energy levels. The energetics. We're going to look at the delta G not prime. Remember, the not means 1 molar concentrations. The prime means 1 molar concentrations for everything but pH. So it's pH 7. And let's look at the kilojoules per mole. We can see that the energetics are highly exergonic for a number of steps. For our pre-citric acid cycle, which is under control. For our first step, that's under control. Our first decarboxylation, that is under control, and our second decarboxylation is under control. Now, this last step, you can see is very highly endergonic. It requires energy, but it's going to move forward because the next step is highly exergonic. when you go back up to number 1. So it's just like water rushing. Overall, between the pre-citric acid cycle and the citric acid cycle, it is very highly exergonic, very negative delta G. So we're flowing water down a big mountain to get to this stuff. So what I want to point out to you is that the steps that are under allosteric control have negative delta Gs. The question was, why will those carbons coming in not leave that first time? And what controls why they're, is that your question? Yeah. And that is basically the mechanisms of the reactions. That's controlling that. And I think, for me, interpreting that, I think there's a reason why we're refreshing with new carbons. We're not keeping the same carbons in that oxaloacetate going over and over again, that we're refreshing the carbons that come in. Okay, this is the only anabolic pathway that we're going to cover. What do I mean by anabolic? Building up, right? So breaking down is catabolic, and it's oxidative in nature, right? We're taking the carbon, going to carbon dioxide. Now, what are we doing with anabolic? It's, we're building up, so it's going to be not oxidative, but reductive. So we're going to be putting hydrogens on. So the Glyoxylate cycle, I picked this one as the only anabolic pathway that we're going to study because it's so much like the citric acid cycle. And I just find it really neat how similar they are. So do you think that we are going to have the decarboxylation steps? No. Why? Why wouldn't you have the decarboxylation steps? It's oxidation. Great. And it's also, you're building up. So why would you lose carbons, right? So we're going to, in this pathway, what we're going to be doing is bypassing those decarboxylation steps. We're bypassing those. We have a couple new reactions. Instead, we're going to take that isocitrate that normally would have been alpha-ketoglutarate, made into alpha-ketoglutarate, and we're going to chop it in half. We're going to chop it to a 4-carbon malate and 2-carbon glyoxylate. That glyoxylate will be put together with another 2-carbon molecule, another acetyl-CoA, and made another malate. I am not going to focus necessarily on the enzymes. We have enough enzymes to remember between the two pathways. I want you to focus on what's different between the citric acid cycle and the glyoxylate cycle and why. Okay? So when you're studying the glyoxylate cycle, what I want you to know is what is different between the glyoxylate cycle and the citric acid cycle and why. Okay, so cool. All right, so normally, this is normal. Oxaloacetate, we just kind of turned our citric acid cycle to the left, all right, counterclockwise a little bit. So this was our start, acetyl-CoA and oxaloacetate, right? Made citrate. We're good with that. Citrate to isocitrate, we're good with that. This is new. Isocitrate is 6 carbons, right? What we're going to do is we're going to chop it. And so on your notes, what I'd like you to do is say this isocitrate is chopped. So this is chopped right here. Chopped to 4 carbons. Say 4 carbons next to the succinate and 2 carbons next to the glyoxylate. So 6 carbons here in isocitrate to 4 carbons succinate, 2 carbons glyoxylate. 6 carbons here, chopped, 4 carbons here, 2 carbons here. All right, so this is a little confusing. draw a circle around your succinate and then do the arrow down. So the succinate is processed as normal for the rest of the citric acid cycle. So what we've done is we've jumped from isocitrate to succinate. We've bypassed, jumped over the two decarboxylation steps and the step that made GTP. Yes, Aaron?
So we don't, for the rest of the process, we don't do anything with succinate?
No, you're going to, that succinate is going to be processed normally. So you're going to do a circle around the succinate, and that is what is going through to the fumarate, the malate, and oxaloacetate. Now then what I want you to do is draw a circle around the glyoxylate and draw an arrow this way. Let me see if I can do that with my pen. So that's what I wanted you to do. So 6 carbons, 4 carbons, 2 carbons. Chopped. So it's broken up, that 6 carbons to 4 carbons. This is the normal citric acid cycle. By going from isocitrate to succinate, we've missed the decarboxylation. All right, it's taking, that's supposed to be decarboxylation. By going from the isocitrate to this step, we've jumped over the decarboxylations. Remember, the decarboxylations got rid of 2 carbons. Well, now these 2 carbons are in the glyoxylate. So instead of decarboxylating, we've kept those 2 carbons in the form of glyoxylate. Instead of losing them as carbon dioxide, we're keeping them. So the isocitrate makes glyoxylate and succinate. The succinate is processed normally, as we do with the citric acid cycle. The glyoxylate we're going to put together with an acetyl-CoA. So that's 2 carbons, plus another 2 carbons, makes our 4 carbons malate. So these are the 2 new steps, this chopped and this condensation. Of acetyl-CoA with glyoxylate. So we're missing 2 and we're gaining 2. So the whole reason for this is to make lots and lots of oxaloacetate, which can go into a pathway called gluconeogenesis. Gluconeogenesis is a way that we can make more glucose. Can we do this? No.
Where do we get the acetyl-CoA from?
Oh, you stole my thunder. Sean stole my thunder. He said, where do we get the acetyl-CoA? Well, we said that this happens in plants and seeds, right? Before we can start doing photosynthesis to make glucose, right? So acetyl-CoA, we're going to find that you can get from the breakdown of fats. And seeds are filled with fat, right? Good fat, but fat. So we're going to get the fat from the, this is how your plants grow in the dark. You're always wondering, like, well, how do plants grow if the seed's down in the ground, right? There's no light to fix carbon dioxide to make glucose from that. Well, it does gluconeogenesis after getting oxaloacetate from the glyoxylate cycle. So it's going to take the acetyl-CoA from the fats. So fats give you acetyl-CoA, and then the acetyl-CoA goes through the glyoxylate cycle to build up more oxaloacetate, and that's siphoned off to make glucose. And so you can get your plants, maybe we'll see, hopefully they're not all dying right now, so in our spring storm, and make our little shoots. Now, once it can start fixing carbon dioxide, this goes away. So this is our first new step. Isocitrate going to succinate and glyoxylate. So these carbons would have normally been lost through carbon dioxide. We're going to keep. Oops. That's not a question mark. That's an exclamation point. Come on. We're keeping those. And we're going to put that glyoxylate together with acetyl-CoA. To make malate. So cool. And so it happens in glycosomes, in plants, and yeast and algae in the cytoplasm. And it helps plants grow in the dark. So seeds are rich in fats, and they use acetyl-CoA from the fats, oxidation, which we're going to learn about later this month. And it produces oxaloacetate for gluconeogenesis. And then once we can fix carbon dioxide, the glycosomes disappear. Kind of cool. I just find it surprising. It's really kind of neat, these things that you learn about. So that's the only anabolic pathway. Now, are there any questions about the glyoxylate cycle?
Fumarate's not produced, it just bypasses?
No, fumarate is part of it. This is when we take the succinate and we process it, fumarate's in there. Because we're going to do the normal processing of the succinate. Succinate to fumarate to malate to oxaloacetate. So those are the same enzymes, the same reactions as the citric acid cycle. What's new is this step and this step that we outline in this slide and this slide. Okay, so the last part, I don't want you to get overwhelmed by my slides because it's busy, but it's busy for a reason. They're to show you how the citric acid cycle is really central to all our pathways. And it's the way how a lot of pathways are related. They're related to each other through the citric acid cycle. So remember, catabolic is breaking down. And then so what we're eating can break down, and they actually can be fed into the citric acid cycle. So it plays a role. So what I want you to think of is that citric acid cycle and all those intermediates in there, they can go in and out. If we need one of them for something else, it can be siphoned off. If we have extra of it, it can be fed in. So when you have extra sugars, fatty acids, or amino acids, they can be processed and broken down so that they can enter into the citric acid cycle. So here's our citric acid cycle. All right, we have citrate, isocitrate, alpha-ketoglutarate, and so on. All of our intermediates. That's what I mean by intermediates of the citric acid cycle. So when I say, okay, you're eating protein, it's broken down into the amino acids, and what you can see is we can break them down and break them down to intermediates of the citric acid cycle. All right, so you can break them down to intermediates and we can feed it in. So this is how the citric acid cycle plays a role in catabolic pathways. Breaking down, you can feed into the citric acid cycle. Do you think that might be like an essay question? How is the citric acid cycle plays a role in catabolic and anabolic pathways? So in the catabolic, you can take fatty acids, you can take sugars, and you can take proteins and break them down to feed in. So here we're showing, and you don't need to know details, just generally answer that question. you can take the, you can see here we have some amino acids. They're making glutamate, which makes alpha-ketoglutarate. All right, these arrows are just different pathways, or these little arrows are pathways. The big arrows is feeding in. Lipids, broken down into fatty acids, and we're going to learn about beta oxidation that will make more acetyl-CoA. We can also get more acetyl-CoA from the breakdown of carbohydrates. So the lipids and the carbohydrates mainly make our acetyl-CoA. Our proteins, our amino acids, can make the other intermediates. And you're just stuffing it in, okay? Building up those intermediates so that we can have the citric acid cycle go forward. If we start depleting those, then it's not going to go forward. I see some complex looks out there. Is there any questions about this? Again, I only want to know generally. So we can use it in an anabolic way by siphoning off for biosynthesis. Oxaloacetate, remember we just said through the Glyoxylate cycle is used in gluconeogenesis. If we take off too much of it, then the citric acid cycle can't go forward, and we need an anaplerotic reaction in order to replenish it. All right, so here I wanted to point out that the anaplerotic reaction to get more oxaloacetate. So remember, green plants and some bacteria can go from acetyl-CoA to oxaloacetate. That's through the Glyoxylate cycle. And then we can take pyruvate to make more oxaloacetate, so that's our anaplerotic reaction. But we cannot take our acetyl-CoA and do that, unfortunately. That's from fats and go to oxaloacetate. That would be very helpful in our metabolism to process more fats, right? So we can't do this. So if we are perhaps siphoning off too much alpha-ketoglutarate to make glutamate, then we need to refill it. Or if we're siphoning off too much oxaloacetate, we can see that we're perhaps using pyruvate or acetyl-CoA to refill it. Here again, we can take pyruvate. We can do this, pyruvate decarboxylate. And if we take off oxaloacetate or malate, bless you, we can go to gluconeogenesis. All right. So we can't make fats into carbohydrates. That's us, right? We cannot make fats into carbohydrates. We don't have the Glyoxylate cycle. That's what plants do. The Glyoxylate cycle is taking that acetyl-CoA from the oxidation of fats and making it into carbohydrates. But unfortunately, we can make carbohydrates into fats. And what we do is we take acetyl-CoA, which we can get by reactions, by reacting citrate with coenzyme A. We can get acetyl-CoA and oxaloacetate. So that's the backwards of the first one. We have the input energy. You can also get oxaloacetate, which eventually produces another other different coenzyme, which is needed for lipid synthesis. So I just wanted to point out that we can make and go backwards with the input of energy to acetyl-CoA, which then we can make into fats. So there's you can build up fats. So this is why when we eat a lot of carbohydrates, we can get fat. Right? We make fats from that. I'm not going to go into it too much, but know that with the input of energy, we can take citrate and make acetyl-CoA. This is a very busy slide, but this is showing how we can siphon off intermediates to make basically the reverse of the catabolic pathways are feeding in, we can siphon off to make our amino acids. We can siphon off to make our fatty acids, which I was just showing, and carbohydrates. So this is, again, showing it in an anabolic way. So catabolic, feeding in, anabolic, siphoning off. Anaplerotic, just replenishing. Now, what we're going to focus on next is how we link the citric acid cycle to the last aerobic metabolic process, the electron transport chain, and oxidative phosphorylation. We take our NADH and FADH2, and we pass their electrons on to oxygen. And so that is what we're going to study next. Did anybody